To make the electrode assembly for multi-channel in vivo recordings, 4 differential electrodes are created and spun together with the individual differential electrodes branching apart at the ends. The anesthetized animal is pinned out, and an incision is made to the side of the eye spot. The target nerves and muscles are located, and electrodes are attached with a drop of superglue at the base of the hook, and then insulated with Quicksil sealant.
Hi, I'm Miranda Cullins from the laboratory of Dr. Hillel Chiel in the biology department at Case Western Reserve University. Today, I'll show you a procedure for assembling and implanting electrodes for in vivo recordings in Aplysia, californica. We use this procedure in our laboratory to study the neuromuscular control of feeding in Aplysia. Let's get started.
To create the electrode, use scissors to cut a piece of enamel-coated 0.001 inch diameter stainless steel wire about 2 feet long. Attach a small ball of putty to each end of the wire, fold the wire in half, and spin the ends to create a twisted pair, called a differential electrode. Tape both ends of the wire to prevent unraveling. Make 4 differential electrodes, using a different color of tape for each differential electrode for identification purposes. Twist 2 of the differential electrodes together, leaving 2 inches untwisted on one end and 1 inch on the other end. Repeat with the remaining pair of differential electrodes.